So when talk about DNA, so again, DNA contains pentose sugar, but in this case, guys, when talk about polynucleotide, you can give a general one, pentose sugar. But when called DNA, you cannot say pentose sugar anymore. Because when talk about DNA, the pentose sugar must be deoxyribose. So highlights here, okay? When asked about this, you have to give a specific one, deoxyribose, phosphate, and 1 out of 4 nitrogenous base or nitrogen-containing base. So talk about DNA molecule. So 2 polynucleotide chain arranged in the double helix structure. Okay, so consists of 2 chains, guys. From the outside of the helix, then the bases belongs to the 2 chains now associated as a pair along at the central axis of the helix. So means that it's something like your ladder. You try to imagine, guys. We have one strand like this. Can you see that? And then you have nitrogenous base. And then we have another strand, nitrogenous base pair up. Are you clear? Forming the double helix. So means this thing structure, the twist. Are you clear? Like ladder, it twists them. So it becomes this double helix. Okay? So exactly 10 base pair are present in the full turn of the helix. Each turn constitutes of 3.4 nanometer of the length. This extra information is for you guys. Okay? Now, something special about the double helix. If you measure the diameter of the double helix, because the double helix turn, right? You can see that it turns. When it turns, it forms something like a cylinder. Can you imagine, guys? The DNA, it turns. It's like paper, you turn it, it becomes a cylinder. Are you clear? So this cylinder, you measure the diameter, is exactly 2 nanometer. Exactly 2 nanometer. Or the radius is 1.0 nanometer. Are you clear? Okay, the diameter is 2 nanometer. Okay, so radius is 1. Now, why they can maintain? Why they can maintain all the way from the first, the top part, all the way to the end? Why they can maintain this radius or diameter? Because of the complementary base pairing. How they base pair? They always pair purines and pyrimidine. Okay. Now, if you look at this, the length of purine is 1.2 nanometer wide. Purine is 1.2 nanometer wide. Pyrimidine is 0.8 nanometer. So they always pair purine with 2 ring and 1 ring pyrimidine. So that's why I can always maintain the diameter 2.0, 1.2 plus 0.8, 2 nanometer. Let's see that. So that's why the complementary base pairing is very, very precise. Are you clear? It's very, very precise. Always purines and pyrimidine. Okay. So the study showed that each base pair, 1 purine, will pair with 1 pyrimidine, which 1 ring. So therefore, exactly 2 nanometer in terms of diameter. By contrast, if you pair 2 purine together, 1.2 and 1.2, then your DNA won't maintain 2 nanometer. It will maintain like this. Can you see that? If you join 2 purine together, 1.2, 1.2. So 2.4. 1.2, 1.2. Or you join 2 pyrimidine together, become like this. Can you see that? So this thing won't happen in your DNA. Now, if I say won't happen, it sounds a bit wrong because it happened. It will happen in our DNA. But the thing here is, you try to imagine your DNA suddenly have a bulging area or something have a groove. Our DNA repair mechanism will repair it. Are you clear? If no mutation takes place. Are you clear? Okay. So complementary base pairing takes place between 2 nucleotide strands. So adenine always pair with thymine, A and T, C and G. Are you clear? Adenine with thymine, A and T, G and C. and they have the hydrogen bond formed between both of them. So it's hydrogen bond that link two polynucleotides together to form D and A. Okay, so A and T, G and C. So how they find out? How they find out this? A always with T, C always with G, actually found out by Chaga. So that's why we have the Chaga rules here. Okay, Chaga rules. So why does Chaga rules actually state that? Chaga rules state that because A and T always pair together, so the ratio A to T, it must be 1:1 or relatively 1:1. Are you clear? So that means by Chaga rules. Okay, and because G and C pair together, when G and C pair together, so means that the ratio of G and C must be 1:1. I cannot have 10G but 5C. How they can pair together? They can't pair together, right? Okay, so now this is the structure of DNA molecule. So each turn, guys, can you see that? Each turn, each turn will be 3.4 nanometer, and the diameter is 1 nanometer. And always you have the G and C, A and T. Okay, C and T, they are pyrimidine. G and A, they are purine. So always purine with pyrimidine. So every turn, okay, you have 10. 1, 2, 3, 4, 5, 6. You have 10s, okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so 2 hidden here. So total 10, they make a turn. Okay. So what Chaga rules actually state that if you look at this, okay, if you look at this, A and T, C and G, definitely you won't get exactly the same because due to the mutation. Okay, sometimes you may have, A pair with others. Okay, so that's why you see that almost 1:1. So when you analyze the ATCG from this diagram or this table, you can see that. Can you see that? And bacterial, E. coli right now is bacterial. Bacterial lack of the proofreading. There's something special about bacterial compared to the eukaryotic cells. Eukaryotic cells, because we have a very tightly regulated outturn of the gene, so therefore you can see that ours one are more to 1:1. You can see that A and T, C and G are very close. But for bacterial, they're slight. Can you see that? Not so regulated. So mutation in the bacteria is very, very high. Virus even more. Are you clear? Okay, so you know that having stuff about the COVID until now we have Omicron, I don't know how many more. Are you clear? So mutation rate is very high in virus and also E. coli, but not the eukaryotic cell. Okay, so you calculate A and T and C and G, you get the ratio about 1:1. Okay, so the simple ones, so 1:1. So we reference the table 10.2, explain how the results support Chaga's rules of complementary base pairing. So Chaga say what? Chaga said that in terms of base pairing, it must be A and T, C and G. Not A and G, C and T. No, it cannot be this way. Are you clear? So Chaga said this. Can we explain by using the result? Yes, can. You can see that A and T always in ratio 1:1, C and G always in the ratio of 1:1. So it means that they must be pairing together. If let's say our DNA is single stranded, do they need to pair? They don't need to pair. Should wait? They do not need to be 1:1 for A and T. Cannot. Are you clear? Okay. So in terms ratio, you can write down 1:1 for O. So regardless of the source of DNA of any species, the ratio of A and T and C and G is always around 1:1. So therefore, the data support Chaga's rules of complementary base pairing. It also shows that adenines always pair with the thymines, so one purines and one pyrimidine. Cytosines always pair with guanines, okay? Against cytosines, pyrimidines, guanines, purine. So the base pair between the purine and pyrimidines maintain the diameter of the DNA double helix. Are you clear? You try to imagine two pyrimidine to form group. Can you see that? It will be detected by the DNA repair mechanism. Even two purine, you're bulging like this. Are you clear? Here. So since we know that complementary base pairing actually takes place, so how hydrogen bonds can be formed. All right. So always remember O is delta negative. Always remember O is delta negative, H is delta positive. Are you clear? O is delta negative, H is delta positive. So means that one delta negative, one delta positive, they form the electrostatic charge. Okay, so look at this, how we can form. So between the A and T, only 2 hydrogen bonds, guys. A and T, 2 hydrogen bonds. Okay, you can try draw out. So I see the A and T, A and T, 2 hydrogen bonds. C and G, 3 hydrogen bonds. So that's why you may have a question asked. Okay, can we have the adenine pair with cytosine? Okay, because also 2 ring and 1 ring. Okay, why thymines don't pair with guanine? Okay, so it depends on what we call this, the formation of the hydrogen bonds and complementary base pairing. So the word complementary means that they complement each other in terms of the shape. Are you clear? Okay, it's something like enzyme and substrate, the shape complement. Okay, so that's why adenine won't pair with cytosine, thymine won't pair with the guanine. So how the hydrogen bonds can be formed? So again, guys, look at this. Okay. H and O, O delta negative, H delta positive. This is how hydrogen bond, first hydrogen bonds is formed between the O and H. Now another one is N and H. Here. Okay, so 2 hydrogen bonds. Write down 2 hydrogen bonds. Between 2 hydrogen bonds between the adenines and thymine. Okay. Then how about guanine and cytosines? Guanine and cytosine, 3 hydrogen bonds. So this is the one, first, second, and third. So student always ask, right? How come this part no? Always remember C won't be electronegative. C won't pull the electron nearer. Are you clear? C atom, the carbon atom here won't pull the electron nearer. Only N and O. Okay, write down only N and O. They are more electronegative, pull the electron nearer to them. So therefore, H become delta positive, O become delta negative. Right? Clear? So 3 hydrogen bonds. So the 2 chains run in opposite directions known as anti-parallel. Now, this is something new that you guys need to know. So each end of the double helix must have an exposed 5 primes, read as 5 prime, phosphate, and an exposed 3 prime hydroxyl group. Okay, on the other. Clear? So what does this mean? Now let me show you guys how this thing actually forms. Okay, how the reaction actually takes place and form the phosphodiester bond first. Okay. Okay, so now let us have the first okay nucleotides here. Okay, first nucleotides. So for any nucleotide, I will start it with the ribose or deoxyribose sugar in this case. Okay, it's a pentose sugar. So if you look at this, carbon number 1 attached to the attached to the nitrogenous base. Okay, so it's a base. I put a B as a base. So this is the deoxyribose, the pentose sugar. Now, if you look at the pentose sugar, if I label them, carbon number 5 always outside the ring. So carbon number 5. So CH2OH. Okay, this is carbon number 5. So where's carbon number 1? Carbon number 1 attached to the base. Carbon number 2, because it's a deoxyribose, so H. Carbon number 3, OH. This is carbon number 2, carbon number 3, carbon number 4, carbon number 5. So reactions take place, okay? Reaction always take place at carbon number 5 and carbon number 3 here. So that's why we have 5 primes and 3 primes. So this end is known as 5 prime ends of the nucleotides. This end is known as 3 prime ends of the nucleotides. I haven't put in the phosphate group yet, okay? I haven't put in the phosphate group. Don't worry about this, okay? Are you clear? Okay, so the top part actually is 5 prime. The bottom part is 3 prime. Are you clear? Okay, these nucleotides. Now, let us start with the first nucleotide. What actually happened here? So I draw it larger. O. So this one connected to the nitrogenous base. And this is carbon number 5. CH2, OH. Okay, so this part OH. So I focus in this part only, guys. I focus in this carbon number 5, carbon number 3 here. So what actually happened now, guys? Look at this. A nucleotide turns this ribose sugar or deoxyribose must react with the phosphate group. So phosphate group is this P, double bond O, OH, OH, OH. Can I see that, guys? Are you clear? So let me bring it down a bit. So reaction actually take place. So how reaction actually take place, guys? Now look at this. Phosphate is acid. CH2OH is alcohol. Cannot OH group alcohol. Right. The OH group here, alcohol. So alcohol donates hydrogen atom. Acid donates OH. So means that reaction actually take place here. So this one remove as H2O. So when the bond is formed, it will form what we call as the ester bond. Sorry, it will form ester bond. Right, alcohol and acid ester bond. So what is the final product here? The final products you get. Okay, then we have the it's black color, and then phosphate. Okay, so the POC bond here, guys, this is C, right? CH2, correct? Not CH2. So POC here is termed as the ester bond. Are you clear? It's ester bond. Now the process haven't done yet. Look at this. This is a nucleotide. I duplicate it and put it here. What actually happened here is, okay, so here is an ester bond, right? POC1 ester bond. Can I see that? Okay, now here we have OH group again. So from the acid donate OH, alcohol donate H. So it formed another ester bond. Can you see that? So I have the first nucleotide, I have second nucleotide now joined together by another ester bond. Do you realize it? Clear? So what is the product formed here? So you can see that, guys, this one removed as water. And then this one formed the bond. So where is my ester bond? This is another ester bond, COP. Can I see that? Another COP. So we have first ester bond. This is another ester bond. So combined together together a phosphate group. So this bond is called as a phosphodiester. Are you clear? This bond is called phosphodiester bond. So if we put it here, it will be COPOC. So this bond, can you see that? COPOC. Two ester bond, one ester bond, two ester bond, so formed together becomes the phosphodiester bond. So phosphodiester bond is the bond that link two nucleotides together. Okay, so when I make it easier later. We can okay, guys, you want to take a photo? You can. Okay. So this is the first one. Okay. Now next. Done. So next. I think somewhere here, right? Okay, you can form the phosphodiester bond. Okay, can? So now, normally we won't draw in detail like this. Okay, we don't waste the time to draw in detail. We will use a geometric shape to represent. So how we represent? We always use a pentagon. Or to represent my deoxyribose, and the shape to represent the nitrogenous base, and something like this with circle to represent the phosphate group. So this is one nucleotide. We always represent this way. Are you clear? Can? So one thing you have to know that phosphate group, if I join another nucleotide here, sorry, I think I draw it this way better. So the bond will form this way. Can I see that? So this is the phosphate. You can see that this part, COPOC. So phosphodiester bond cannot. Okay. So you can see that if I continue like this part will be free end. And this one, OH group, will be free end. Cannot free end. Are you clear? So this free end indicate what? Because this is the 5' carbon. So because of this free end, phosphate group attached to the 5', so this end is termed as a 5' end. Are you clear? Because attached to who? Attached to carbon number 5. The free end, nothing, right? In this case, after phosphate, anything else? Nothing. So this is free end. So the free end here attached to carbon number 5. So therefore, it's termed as 5' end. How about this? Now, next, OH group is a free end here, right? Nothing. No one joined. No one joined the OH group, right? So OH group joined to carbon number 3. So this part is a 3' end. 3' end. Are you clear? So what's meant by anti-parallel? What's meant by anti-parallel? Anti-parallel means that if you look at this, if you read it from top down, it's a 5' to 3' cannot. The opposite strands top down will be 3' to 5'. So it will be opposite. Okay. Duplicate. Then how many go rotate? So complementary base pairing will be this way. Can you see that, guys? So I remove. So this is the first strand. this is a second strand, cannot. They form the hydrogen bond. Let's say A and T, C and G. But they are parallel, but anti-parallel, because you can see that this is a 5' to 3' top down. Now here, look at this OH group. OH group, this is the free end, cannot. So it's a 3', carbon 3. This one, phosphate, is a free end attached to carbon 5, so 5'. So now top down become 3' to 5'. So this is termed as Are you clear? Okay. Now, can you see that? So this is termed as anti-parallel. Okay. So double strands. Okay, double stranded. Okay, so with this, go back to our notes as well. Sorry, guys. So basically, from here you can see that, okay, OH group again attached to the carbon number 5. OH group, not OH group, the phosphate group attached to carbon number 5, OH group attached to carbon number 3. So this 5', 3' on an opposite, 3', 5'. Are you clear? Okay. And C and G, 3 hydrogen bonds, A and T, 2 hydrogen bonds. Okay, so this is a structure known as anti-parallel. Okay, so with this, I've done for the DNA structure.